So we are back again. So this time around, we are going to look at basics of chemical structure, the representations, and how we bring them alive. Draw them, right, using some basic tools. So as consistent with the goals of these tutorials, it is just a refresher. As we said, very useful for undergraduates and those in the entry level of postgraduate level. To just pick their interest, draw them in on the most fundamentals that are necessary for them to launch their careers. Okay. So we're going to run through this: chemical structure, protein structure, file formats, representation, conversion, drawing them, and all that. Meanwhile, I'm Shadrach Eze, and then let's go. Okay, so what's the chemical structure? So this is a structure before us. So the question is, how do we represent this structure intelligently in a way that is realistic and in a way that truly model the physical representation in real systems? Okay. And then second question will always be, how do we communicate this representation? So we'll deal with representation, we'll deal with communication when we come to file systems. Okay, so we can either check the zero dimension, which is okay for each particular, let's say for carbon, oxygen, whatever, they're in a point in space, that is a zero dimension. So you can locate them at a particular point in space. Now, at one dimension, maybe this carbon is connected to this nitrogen, that is just a single line on the flux. So that is a one dimension. So you can say, oh, so that's why you're looking at substructures. So carbon is connected to nitrogen, oxygen is connected to so. If we now try to build the whole system, we're talking about two dimensions. So we'll be able to connect the whole structures together in two dimensions. Now in three dimension, because two dimension is still on the paper, so it's still flat. In three dimension, we are trying to build it up in 3D space in XYZ coordinates. So how does this structure look alike, or how does it look like in the 3D space? And that is actually what the 3D is all about. Now, because as you look at this structure, it's just flat on the paper. But is this carbon lying exactly close to nitrogen in real life? The answer is no. So the 3D structure gives us that. Now for the representation, is just like an advanced 3D, but you are not talking about, let's say, enclosure, when you box a system, solve it. So we're not talking about the things that also interact with the molecule. We talk about 4D. Okay. Now, as we move from 0D to 4D, the richer the information, but the more difficult it is to compute. Computational resources, it cuts down a lot. Now, just a refresher, as I mentioned to you about proteins. Remember that proteins have 4 kinds of structures, classes: the primary, the secondary, the tertiary, and the quaternary structures. Okay, so we just talk about the chain, then the alpha helix, beta sheets, and all the terms and everything for secondary structures. Tertiary structures, they fold into globular proteins, whatever, and all that. And quaternary structure, we're talking about maybe 2 or more monomers combining together to form very big shapes. Now, and then the basic unit obviously is the amino acid. Then for lipids, we remember lipids have a hydrophilic head and hydrophobic tail. These things are just basic, but they are really important in molecular modeling, because how you build system for protein is very from how you build the same system for lipids. If you are going to maybe simulate it or try to model real life events. And then for DNA, obviously, made up of the backbone, which consists of the bases connected together using hydrogen bonding, in base, so phosphate backbone, then attached to the bases connected together using hydrogen bond. So this is just like basics. Okay. Now let's talk about let's zoom into the protein itself. Now what I'm going to explain is very important, because how you interact with your protein, especially protein, because we don't work on maybe DNA for now, work mainly on protein. How you interact with it depends on your understanding about the structure of the protein at the basic level. I'm talking about the amino acid level and the peptide level. So we know that for each protein, there is an alpha carbon, which is like the central carbon, connected to a nitrogen and a carbonyl group. Then at that alpha carbon, there's a side chain. You can see side chain 1, side chain 2, side chain, so the side chain. So we know that it's only glycine that does have a side chain because it has the hydrogen at the side chain. So when you are looking at this amino acid, so and then it's important to know that the bond between this nitrogen here, carbon here, and this carbonyl group is stiff because it's an imide bond, right? It is stiff. And then most motions that this residue is going to undergo will be heavily dependent on the flexibility of the side chain. That is important. And that is what is illustrated here. You can see the alpha carbon at carbon number 2. Yeah, sorry about that. You can see the alpha carbon at carbon number 2. You can see the nitrogen here, N1. You can see the other carbon 2, the carbonyl carbon here. And you can see the beta carbon, which is already an extension of the side chain. So this is a cytosine, as you can see. And then the flexibility will determine wholly on this side chain. So most times the backbone doesn't move, although it does. Maybe sometimes it rearranges from, let's say, from a coil to a sheet or whatever, but it's very rigid and we have to bear that in mind. And then as you can see here, these extensions of these side chains are the most important things when we discuss fluctuations in protein level or amino acid level. And most of these things are critical in the binding of ligands, in allosteric communication, and whatever. Yeah. Okay, so let's look at chemical representation. So first of all, how do we represent a molecule? If you say, oh, this is paracetamol, this is indomethacin, this is morphine, how do you represent it? Obviously, generic names, or paracetamol or morphine hydrochloride, as you can see here. Molecular formula, the molecular formula of methane is CH4. That is very intuitive and very informative, but not so powerful. Then you can also come about the IUPAC name, which is very lengthy. Do you understand? Anyway, can computer understand IUPAC name? Yes, but very complicated. Then we'll pass register number. So we're not dealing about some database and how they store them, because every database, when they store a molecule, they give them a number. Talk about PubChem, talk about ChemBuild, they give it a number. So what is that number? That number usually is distinct, but does it tell us every detail about the molecule? The answer is no, because 6, 7, 9, 2, and 5 has nothing to do with dichlorovarin hydrochloride, right? So we now come down to the SMILE notation, the INCHI, and all along. So these are, you see, these are increasing complexity of how we can encode the information available in a molecule so that the computer will understand it on one hand, and they will be able to fully represent the molecule on the other hand. So let's talk about this SMILE, the Simplified Molecular Input Line Entry System. So it's just a basic way of counting the heavy atoms. So for example, the hydrogens are excluded. So how do you count, how do you represent the heavy atoms of a molecule while maintaining their connections, how they're connected to each other? So for this particular molecule, we work from nitrogen here in this piperidine, and then the nitrogen N1, then we walk back to C1, which is here. So you see N has 1 and C has 1 because they form a ring. So it means that if you start at N1, this C1 also has to connect to N1 because that is a ring. And then you walk, and that's how you build this molecule. So if you want to know more, there are links at the end of the tutorial. You can learn more how to generate whatever SMILE notation you're interested in. Now, the most important thing about this SMILE notation is not just how to generate them, is that anybody can start anywhere. It means I can start at this 3, I can start at this 0. So people have 1001 ways to represent a particular molecule. And that's why, we've generated canonical SMILEs. There's a concept of canonical SMILEs, which has been implemented by IUPAC, where the SMILE notation has to start from somewhere at a particular point in time, usually based on the IUPAC numbering, so that we can standardize it. And that's very important in machine learning, molecular modeling. Converting normal SMILEs to canonical SMILEs is very important so that you standardize it. If not, you will end up having you end up having the same molecule appearing 10 times in your data set. But because their SMILE notation were generated from different points of the same molecule, they are obviously different. Your database can't differentiate them using only SMILEs, right? Okay. So why SMILE notation? They are very flexible, computer readable. Yeah, because they're just sequences of strings. They're strings of alphabets and numbers, so computer can read it. They are universally accepted and they encode deep information. For example, they encode double bonds, they encode even aromatic groups, they encode rings, everything. So it's only the hydrogen that is not there. So they are very, very depth. They have depth. Okay, now let's talk about SMARTs, which is SMILEs arbitrary target specification. So if you have a database and you want to select all the component database that has phenyl group attached to them, how do you go about it? And so you see, so this is a way of searching databases. It's based on smart signals or smart patterns. So it means that we can find a way to encode a general pattern or a general way of representing the the phenyl group wherever it is found in a molecule, and it's based on SMILE, so it's not very complicated. So you can see here, this is the implementation from RDkit. RDkit import chem.move from smart C1 CCC1 #6. So it means this is a benzene ring, obviously, because it starts from small letter C1 to small letter C1, which is 6. So this is a benzene ring. But we are looking for a benzene ring that is attached to a carbon atom because it uses the molecular weight of the atom, 6 for carbon, 8 for oxygen. So it attached to carbon atom. So it should find all the molecules that has a benzene ring attached to carbon. So maybe a methyl group, whatever. Let's see what it found. Maybe just a snapshot, not everything. So you can see it found a methylphenyl, or what we call toluene or something. Then it found a biphenyl. It also found an this it also find this multi-ring system. Do you understand? Yeah. So this is an indole. So it means that we can literally use that pattern to query our database and find out all the molecules that match that particular pattern. Okay. So these are additional ways of specifying whatever you want. Do you need only single bond, double bond, triple bond, or whatever? Do you need aromatics, any kind of bond? So these are detailed information, but this is only relevant in querying databases and finding substructures in your molecules, right? Yeah, because I mean, it's not a very regular, it's not a everyday, not for everyday use. So, but if you're dealing with databases, finding substructures and all that, it's very useful. Now, let's talk about INCHI. INCHI is one of the advanced way of representing chemical structures, yeah, because it can encode stereoisomerism, can encode charges, terrible things. It's just that it's complicated and very, very long, right? So, as you can see, the INCHI version, the chemical formula sublayer, hydrogen atom sublayer, stereochemical layer, everything is available. Charges are available. So it encodes everything. And then we even have an inch key because of the length. IUPAC has denoted an inch key, which is a way to encode the inch so that it will be shorter, computer readable, but it's still not like a very popular tool in machine learning, no, but it's a standard tool, obviously, very standard out there. Okay, now let's talk about file format. We just finished talking about chemical representation and molecular representation. So what is file format? So don't forget, molecular representation is a way we model the structure of a molecule. Do you understand? In, let's say, spatially, in 3D, in 2D, how they are connected, whatever, maybe using SMILEs or whatever. For example, you see, SMILE notation doesn't tell us about the 3D structure, but it tells us how they are connected. It can tell us the 2D structure because it's easy. It gives us the bonding and the connection. Now, but that is representing the molecule. Now, how do we communicate this molecule? If I have a molecule of paracetamol and I want to write it down, should I draw the molecule? Do you understand? So how do I encode this structure into a file and send it to you, and you'll be able to decode it? So it's a way to write the data and exchange it and decode it and whatever. So that's what is called file format. And then there are so many of them, 8,000 of them. So we can start with mode, XYZ, just the coordinates, CIF, PDB, and on and on and on. There are a million. In fact, they are proprietary. I mean, if you have a software, most times you design a file format that is suitable for your software. So there are a million of them out there. Now, so let's just look at a few of them. PDB file is just a very simple file. So this is a PDB file of a protein, as you can see. So there are atom headers, atom rows, and then it's, atom column here, but it appears in every row. So it means that these are the specifier for the residues. So atoms for the residue, heteroatoms for ligand, for water, whatever. So then it will give us the name of the element, the amino acid, that is the residue, the chain name, maybe L or 1, 2, depending on what you name the chain, the sequence number, that's also important. Yeah. And then the sequence number, you can see, is very different from the initial numbering. The initial numbering is atomic numbering. I mean, it is a numbering of the atom. So N is the atom number 1, CA atom number 2, and all that. But the sequence number has to do with, because all these atoms still belong to aspartate, so it's still residue 1. That is sequence number, is still residue 1. And then you now have the coordinates. Sometimes you can also encode charges, encode so many other things, but at least the XYZ coordinates. So that's it. Now, PDBQT file is also very similar. It's an extension of PDB file with charges and torsion. As you can see here, there's a charge, there's a torsion attached to it, and then so it's an extension. It's very useful. It's very popular for Vina, Autodoc, and other ones that developed it. Okay. Now, how do we convert between files? So if a file system is a way we encode information about molecular representation, communicate it, exchange it with others, decode it, read it. So how do we do this conversion? Two popular tools. RDkit is implemented in Python. Very, very important, versatile, useful. Then OpenBabel too is available. So OpenBabel can do a whole lot of conversion. So using different force field, you can do optimization. There is a whole lot to do with OpenBabel. It's a whole field of its own. Okay, now let's just talk about OpenBabel. RDkit, we'll be using it along the line when we do some things, but let's just talk about OpenBabel. So how do you install OpenBabel? So OpenBabel has a graphical user interface, so you can run it normally on Windows. But since we are dealing with command line, if I want working on Linux, on Unix, on Ubuntu, on Debian, whatever, we want to stick on the installations on the command line. So sudo app update, you update your utilities, then you install OpenBabel. Okay. Then you can also conda install OpenBabel. You can also install it from the conda forge channel. Then help. To see so many functions available, you can either use the single dash with capital H or double dash with small help to see all the utilities available in OpenBabel. Then you can now run the OBabel, just call it OBabel IPB. So you can specify the input file format. If you don't specify, then the default will be the extension provided here. You can also indicate the output file format. If you don't, then the extension you provided becomes the default extension. So there are so many other utilities available here that you can use. OpenBabel, you can use as we discussed during the basic Linux class. You can also use asterisk wildcards so that it loops over a series of files. So if you have like 2,000 structures to convert, you don't need to load them. Just say, oh, do this, then sit down and watch it happen. Okay, now, what if we want to draw chemical structures? These are available tools we can use. This is Marvin Sketch. It's not open source. You need a license, but usually there are licenses for academic users. This is ChemSketch, open source. but not every utility is still available. You still need to buy some things. Then CameDraw, commercial, you need to have a license. The same thing, Marvin Sketch. Okay, so most of these tools are implemented in most programs. For example, if you are working on uh, if you're working on some site or some especially open source programs online, you see some of these drawing tools implemented in them so that you can easily draw your structure. So you must not download them. And also Python is also here to allow us, RDkit here, as we mentioned, is also here. It's available. It can help us convert from strings to 3D representations or so many things. So sometimes you might not need a chemical drawing tool to be able to draw your structure, but some other times you need them, especially when you want to build a molecule from scratch. And then there are specific things you want to look at. So because sometimes you want to build a molecule from scratch, and then you need some of these softwares to do that. Okay, these are so many of the resources. The initial publication of OpenBabel by O'Boyle, and then practical chemoinformatics, yeah, by Pat Walkers, very very important, and then so many other resources, especially from the maybe Daylight company and all that. So they are very useful. So this is just a brief introduction. As I always say, this is never, cannot be exhaustive. The idea is to refresh your mind and then to draw you in to see that there are so many things you can do, there's so many things available. What are the gaps that are currently in your knowledge? How do you feel them? Yeah. Thank you for joining me.